Inside your cells right now, there's a microscopic machine performing its specific job 40 million times every single second.
Which is just, a completely wild number to try and wrap your head around.
Right. It really is.
Yeah.
And today, our mission on this deep dive is to unpack a fascinating stack of anatomy and physiology sources. We really want to understand exactly what that machine is and how it fundamentally keeps you alive.
Yeah, because we are talking about enzymes.
Exactly. Now, if you're listening to this, you might vaguely remember enzymes from, like, high school biology.
Oh, sure.
You probably think of them as just digestion juices, right? Something floating around in your stomach that just breaks down a cheeseburger.
Yeah, that's the classic textbook definition most people hold on to.
But the reality of what these molecular machines actually are and the physical mechanics of what they do goes so far beyond just digesting lunch.
It really does. I mean, calling them digestion juices is kind of like calling a supercomputer a fancy calculator.
Yeah, that's a good way to put it.
The sheer scale and the speed of the operation is what changes the paradigm entirely. We are looking at a system that executes millions of chemical reactions per second.
Right.
And it does this at the atomic level without generating enough heat to, incinerate the surrounding tissue.
Okay, let's unpack this. Because before we get into how on earth something can physically move that fast without tearing itself apart, we kind of have to look backward.
Right, the history.
Yeah, how did we even figure out these invisible machines exist in the first place?
Well, we have to step back to the mid-1800s. It was a time when our understanding of biology was much less about mechanics and much more about magic.
Magic.
I mean, the accepted scientific terminology of the era was vitalism, but it essentially meant magic.
Wow.
Yeah, brilliant scientists, including Louis Pasteur, argued that certain chemical processes like fermentation required a vital life force.
Huh. So they thought life itself was the active ingredient.
Exactly. Pasteur firmly believed that you could not separate the chemical reaction of fermenting yeast from a living yeast cell. To him, and honestly to the entire scientific establishment at the time, the living cell itself was the magic required.
Meaning the chemistry is just a byproduct of life? Like if the cell dies, the magic dies with it, and the chemical process simply stops.
That was the absolute consensus. But around that same time, other researchers were, trying to categorize the actual substances inside these cells.
Okay.
In 1878, a German physiologist named Wilhelm Kuhne coined the term enzyme.
And where does that word come from?
It derives from the Greek enzyme, which literally translates to in yeast.
Oh, wow. I never knew that.
Yeah. He used it merely to describe the catalytic substances inside the cells. He was just trying to give a name to whatever was doing the work, distinguishing it from the whole living organism.
But it wasn't until a couple of decades later that the whole idea of a magical vital force was actually debunked, right?
Right. It happened in 1897, and it was a complete and utter accident.
I love when massive scientific breakthroughs are just accidents.
It's the best. So a German chemist named Edward Buchner was trying to preserve yeast cell extracts for medicinal use.
Okay.
He and his brother used a mortar and pestle with sand to physically grind up the yeast cells. They totally destroyed them, obliterating any trace of a living cell.
So no vital life force left.
None, just cell juice. And to preserve this cellular juice, he added a highly concentrated sugar solution, which was a standard preservation technique at the time.
That makes sense, yeah. I mean,
Yeah.
you dump a ton of sugar into fruit to preserve it as jam. You just make the environment hostile to bacteria.
Exactly. The intention was just to let it sit passively on a shelf. But instead, the mixture started bubbling wildly.
Oh, yeah.
Yeah, it was fermenting. Buchner had this flask with absolutely zero living cells present, yet the complex chemical reaction was happening right in front of him.
So the juice itself was doing the work.
Yes, he had accidentally proven that you didn't need a vital life force. The chemical activity was coming from specific non-living molecules floating in the juice.
That is incredible.
He called this active substance zymase, and he actually eventually won the 1907 Nobel Prize in Chemistry for that discovery.
That's like trying to invent a new food preservative for your pantry and accidentally disproving magic in the process.
It really is. It's widely considered the founding experiment of biochemistry.
Because it shifted everything to physical mechanics.
Exactly. Buchner proved that life's processes could be studied purely as chemistry and physics, not mysterious forces.
Right.
And for over a century after that, the ironclad rule in biology textbooks was that all enzymes were proteins, period.
Just proteins.
Yep. Proteins were thought to be the only molecules structurally complex enough to fold into the intricate 3D shapes required to act as these molecular machines.
Until the 1980s when the textbooks had to be rewritten again.
Right, again. In 1982, Thomas Seike's lab was studying a single-celled organism, and they observed something impossible.
What was it?
A piece of RNA, which was supposed to be nothing more than a passive genetic messenger, was actually cutting and rejoining its own strands.
Wait, on its own? No proteins involved at all?
None at all. And around the exact same time, Sidney Altman found that the RNA component of another cellular complex was also catalytically active.
Wow.
They called these RNA enzymes ribozymes, and they shared the Nobel Prize for it in 1989.
I know this was a massive deal, but for anyone who hasn't taken a biology class recently, why was discovering an RNA enzyme such a huge shock to the system?
What's fascinating here is that this discovery fundamentally rewrote our understanding of the origin of life itself.
Oh, really? How so?
Well, before ribozymes, biology had a massive chicken and egg problem. DNA is the blueprint, and proteins are the machines that build things.
Okay, makes sense.
But you need the protein machines to read and copy the DNA blueprint. So which came first in the primordial soup? The blueprint or the builder?
Oh, I see. You can't have one without the other.
Exactly. The discovery of ribozymes gave us the RNA world hypothesis. It suggested that early life could have run entirely on RNA.
Because RNA can do both jobs.
You got it. It can store genetic information like a hard drive, and it can act as a physical enzymatic machine to build things.
That is wild. Okay, so we've moved from magical life forces to accidental yeast juice, all the way to RNA kickstarting life on Earth.
Quite a journey.
Seriously. But let's look at the actual physics of this. We know they are machines, but how do they actually speed up reactions?
To understand the mechanics, we have to look at activation energy.
Okay, what's that?
Every chemical reaction, even one that ultimately releases energy, has to overcome an initial energy hurdle first.
Right.
Imagine a heavy boulder sitting in a shallow crater at the very top of a large hill. Even though gravity wants to pull that boulder all the way down to the bottom of the valley, you still have to put in a little bit of initial physical effort to push it up and out of that shallow crater first.
Okay.
That initial push, that little bit of effort, is the activation energy.
Wait, if they don't actually supply energy to the reaction, how are they getting the reaction to happen so fast at just 98 degrees? Are they basically just digging a tunnel through the energy mountain instead of pushing the boulder over it?
Yes, that is a fantastic way to visualize it. They are basically digging a tunnel.
Okay, that makes so much more sense mechanically.
Right, and it's a crucial distinction. Enzymes are biological catalysts. They do not add energy to the system, and they do not make an impossible reaction possible.
You just lower the barrier.
Exactly. They simply lower the activation energy. Physically, they do this by grabbing the molecules involved and contorting them into what's called a transition state.
So they physically grab them.
Yeah. Think of holding a thick wooden stick. If you just press on the ends, it's hard to break. But if you bend it over your knee, you put intense physical stress on the structural bonds in the middle of the wood.
Oh, I see.
Once it's bent like that, it takes just a tiny fraction of extra energy to make it finally snap. Enzymes bend molecules over their molecular knee. By lowering that energy barrier, reactions that would normally require high heat to occur can suddenly happen in milliseconds at our normal body temperature of 98.6 degrees.
And they are highly specific about which sticks they break over their knee, right? Like they don't just grab anything floating by.
Very specific. This comes down to their three-dimensional shape. Every enzyme has an active site, which is a highly specialized geometric pocket.
A pocket, okay.
Yeah, and the physical shape, the electrical charge, the microscopic acidity of that pocket, it all only fits one specific target molecule, which we call the substrate.
Oh, so that's why they're named after their target.
Exactly. This specificity is why enzymes are usually named after their target, just adding the suffix ase. Lactase breaks down lactose, protease breaks down proteins.
DNA polymerase builds DNA.
Spot on.
But how rigid is that fit? Because I recall older textbooks describing it as like a rigid key going into a metal lock.
Emil Fischer proposed that lock and key model back in 1894. He thought the enzyme was a hard, inflexible shape and the substrate fit into it perfectly.
But that's not right.
No. In 1958, Daniel Koshland proposed the induced fit model, which we now know is how it actually works.
So it's less like a metal lock and more like a glove.
Think of it exactly like a leather glove. The active site is somewhat flexible. As the target molecule enters, the enzyme actually shifts and changes shape slightly to mold itself tightly around the substrate.
The glove wraps around the hand.
Yes. That structural shifting is what puts the physical stress on the molecule to bend it.
Okay, but if they are constantly grabbing molecules and, contorting themselves to bend chemical bonds, do they wear out? I mean, if I bend a piece of metal back and forth a million times, it eventually snaps.
That's a super common intuition, but enzymes operate on atomic rules. They are infinitely reusable.
Infinitely. Right.
Really, once they facilitate the reaction and release the finished product, the enzyme snaps right back to its original resting shape, perfectly intact.
Just ready for the next one.
Instantly ready to grab the next molecule. They are not consumed in the reaction at all.
Which brings us back to that unbelievable number from the beginning of our deep dive. The machine doing its job 40 million times a second.
Right. We are talking about catalase.
Catalase. Okay.
This is an enzyme whose specific job is to break down hydrogen peroxide. Hydrogen peroxide is a highly toxic byproduct of your body's normal cellular metabolism.
Oh, so your body just makes it naturally.
It does, and if it builds up, it literally shreds your cells.
Okay.
So catalase grabs that toxic peroxide and breaks it down into completely harmless water and oxygen.
Oh, wait. If you've ever poured hydrogen peroxide on a cut and watched it foam up, is that what's happening?
That is exactly what's happening. That bubbling is the catalase in your damaged cells rapidly turning the peroxide into oxygen gas.
That is so cool.
And a single molecule of catalase can execute this reaction roughly 40 million times per second.
The physical speed of that, the molecules colliding, bending, and releasing, is just staggering.
It's mind-boggling, and catalase isn't an anomaly. Take carbonic anhydrase, for example.
What does that one do?
When your cells produce carbon dioxide, it has to be converted so it can be transported in your blood back to your lungs to be exhaled. Carbonic anhydrase handles that conversion at a turnover rate of about 1 million reactions per second.
A million times a second. Still incredibly fast.
But carbonic anhydrase is interesting because it requires a helper.
Like a sidekick for the molecular machine.
Exactly. In biology, we call them cofactors or coenzymes. Many enzymes are just complex tangles of raw protein, and they are completely useless on their own. They need a metal ion like zinc, iron, or magnesium, or a vitamin-derived molecule to slot directly into their active site.
They kind of turn the machine on.
Yes, essentially. Carbonic anhydrase requires a zinc ion to function. Without that zinc nestled in its pocket, you do not exhale CO2 efficiently.
So these machines are incredibly fast and infinitely reusable, but they also seem pretty fragile if they rely on all these specific metal ions and conditions to work.
Fragile is the defining characteristic here. They operate in strict Goldilocks zones.
Goldilocks zones, like not too hot, not too cold.
Exactly. They need the ambient temperature and the pH to be just right. Heat is just atomic vibration, right?
Right.
So if you raise the temperature slightly, reactions speed up because molecules are vibrating and bouncing into each other faster.
Which is good.
Until it's not. If you raise it too much, the enzyme molecule vibrates so violently that the delicate chemical bonds holding its intricate 3D origami shape together just snap.
Oh, wow. So it just falls apart.
The protein physically unfolds. We call this denaturing. Once that highly specific active site loses its shape, the glove can no longer fit the hand and the enzyme is dead.
That perfectly explains why a high fever is a medical emergency. Like if your internal body temperature gets much above 104 degrees Fahrenheit, your molecular machines are literally physically unraveling.
Exactly. The factory starts breaking down. And it's not just temperature, it's the chemical environment too, the pH.
Oh, right. Acidity.
Think about the enzymes in your digestive tract. You have pepsin, which digests proteins in your stomach.
And the stomach is super acidic.
Right. Your stomach has a pH of about 1.5 to 2, which is basically battery acid. Pepsin evolved to fold into its perfect working shape only in that highly acidic environment.
Okay.
But just a few inches away, in your small intestine, you have another protein-digesting enzyme called trypsin. The intestine is mildly basic, with a pH around 7.5 to 8.5.
If you took trypsin out of the intestine and dropped it into the stomach acid, it would be like taking a deep-sea submarine and dropping it into a volcano. It's a highly specialized machine, but in the wrong environment, it's instantly crushed.
Exactly. The protein would denature instantly. The acid would warp its structure completely. It shows how the exact same category of Proteus is custom-tuned for its specific location in the body.
But wait, if we have these indestructible as long as they stay in their Goldilocks zone machines running millions of times a second, what stops them from just running out of control?
That's the big question.
Yeah, because if I have a machine building things a million times a second, my factory's going to overflow.
This brings us to the control room of the body. What we casually call metabolism is really just a vast interconnected network of these enzyme pathways.
Like an assembly line.
Exactly. Product A is turned into product B by one enzyme, and then product B is handed off to the next enzyme to become product C. To prevent the factory from overflowing, the body has to regulate the enzymes themselves, acting like valves on a pipeline.
How do you turn off a microscopic valve?
One of the most elegant discoveries in biology was made in 1965 by Jacques Monod, Jeffries Wyman, and Jean-Pierre Changeux.
What did they find?
They figured out that enzymes often have a second binding site located far away from the active site called the allosteric site.
Like a hidden off switch on the back of the machine.
Precisely. And plugging something into that off switch triggers what we call feedback inhibition.
How does that work?
Let's say our pathway is an assembly line making an amino acid. When the cell finally has enough of that amino acid, the extra molecules float over to the very first enzyme in the pathway and plug into its allosteric off switch.
Oh, so the product itself shuts down the line.
Exactly. Physically, this acts like pulling a lever on the back of a mechanical claw. It pulls the internal gears out of alignment, warping the shape of the active site on the other side of the molecule just enough so the substrate no longer fits.
That is brilliant. The end product literally loops back and physically deforms the assembly line to shut it off.
It's a completely self-regulating physical system.
But what happens when that regulation or the machine itself fails? Because the stakes seem incredibly high.
The stakes are quite literally life and death. Because the system is a chain reaction, missing even one single type of enzyme out of the thousands in your body can cause the entire assembly line to back up with catastrophic results.
Do you have an example of that?
Take Tay-Sachs disease. It is a fatal condition in infants caused by the absence of one single enzyme, hexosaminidase A.
Just one enzyme.
Just one. Without it, the body can't break down certain fatty substances in nerve cells. The toxic material builds up, causes progressive neurological damage, and it is entirely because one tiny molecular machine is missing from the factory floor.
It really highlights how precarious the whole network is.
Another classic example is PKU, or phenylketonuria. It's a deficiency in an enzyme called phenylalanine hydroxylase.
And what happens without that one?
Without it, a common amino acid from your food builds up to toxic levels, which causes severe intellectual disability. Because the consequences are so dire, medicine stepped in. It's the entire reason babies get a heel prick blood test.
Wait, that standard heel prick test every newborn gets before leaving the hospital is just looking for a single broken molecular machine.
That's exactly what it is. It's one of medicine's most successful genetic screening programs.
Wow.
If we know the enzyme is missing right at birth, we put the child on a highly restricted diet, preventing the precursor molecule from ever entering their system. They can live a completely healthy life because we bypass the broken machine entirely.
Here's where it gets really interesting. I was reading the notes on methanol and antifreeze poisoning, and I want to make sure I understand the mechanics of this treatment.
Okay, let's hear it.
So, if a dog or a person ingests antifreeze, the literal life-saving medical antidote is to flood their system with regular ethanol alcohol. Like, we're essentially keeping the enzyme busy by getting it drunk so it ignores the poison.
Mechanically, yes, that is absolutely right. We are exploiting enzyme competition.
That's so crazy.
The enzyme in your liver that breaks down the alcohol in beer or wine is called alcohol dehydrogenase. But that enzyme isn't perfectly exclusive to ethanol. If it bumps into methanol wood alcohol or ethylene glycol from antifreeze, it will eagerly grab them and process them instead.
But the products it turns them into are worse, right?
Much worse. The antifreeze itself isn't actually what kills you.
Really?
Yeah. The problem is that alcohol dehydrogenase turns the antifreeze into oxalic acid, which forms crystals that destroy your kidneys. Or it turns methanol into formaldehyde, which can blind or kill you.
Oh, jeez.
The poison is what your own enzyme manufactures. So the clinical antidote is to administer a massive dose of standard ethanol or a specific inhibitor drug called fompazole.
So you just flood the zone.
Exactly. By flooding the system with a harmless substrate, the enzyme gets entirely occupied processing the safe ethanol, leaving the antifreeze untouched, so your body can eventually just flush it out harmlessly through your urine.
It's wild that we can chemically trick enzymes in an emergency like that.
It's life-saving chemistry.
But that same ability to manipulate these machines isn't just for life or death hospital visits, right?
Yeah.
We actually hijack them for everyday household chores, like look at your laundry detergent,
Oh, absolutely. Modern detergents are packed with engineered proteases and
to
lipases.
break down proteins and fats.
Exactly. Instead of using boiling hot water to wash clothes, we drop in enzymes designed to hunt down and digest protein and fat stains in cold water.
My washing machine is basically digesting my clothes clean.
That is physically what is happening, yes.
And it goes further than that, even into basic over-the-counter pills. Like, if you are lactose intolerant, you don't really have a disease. Your body just naturally stopped producing the lactase enzyme after childhood.
Right, which is very common.
So those pills you buy at the pharmacy are just capsules packed with the missing molecular machines to do the work in your stomach.
Exactly. Or look at modern medicine. The COVID-19 antiviral drug Paxlovid is an enzyme inhibitor.
Oh, I didn't realize that.
Yeah, when the virus enters your body, it requires a specific protease enzyme to cut its own long protein chains into smaller pieces to replicate. Paxlovid goes in and chemically jams the active site of that specific viral machine, completely stopping the virus from reproducing.
The source notes also mention something about PCR testing and Yellowstone hot springs.
Ah, yes. Polymerase chain reaction, or PCR. It's essentially a biological photocopier for DNA. We use it for crime scenes, genetic testing, viral swabs. To copy DNA in a lab, you have to heat it up to separate the double helix strands. But as we discussed earlier, high heat would normally denature and destroy a human polymerase enzyme.
Right, the factory breaks down.
So scientists went to the boiling hot springs of Yellowstone National Park, found bacteria living happily in the boiling water, and extracted their enzyme Taq polymerase.
Oh, wow.
Because that enzyme evolved in boiling water, it survives the high heat of our lab machines perfectly.
It is wild to think that the exact same mechanical principles happening in a boiling hot spring in Yellowstone are what we use to solve crimes with DNA, or to design a pill to fight a global pandemic,
It's all connected.
or even that common fizzle reaction some people get when drinking alcohol.
Oh, the Asian alcohol flush.
Yeah.
A large portion of the East Asian population has a tiny genetic variant in an enzyme called ALDH2.
And what does that variant do?
The structure is slightly altered, making the machine a bit sluggish. So a toxic intermediate byproduct of alcohol builds up faster than it can be cleared, causing a rapid heartbeat and flushed skin.
Just because the machine is a little slow?
Yep. It's a visible, everyday demonstration of enzyme genetics at work in hundreds of millions of people.
So we started this deep dive looking at how scientists used to believe in a magical vital force. Then Edward Buckner accidentally proved with a batch of yeast juice that life's chemistry is driven by non-living molecules.
Right.
We saw how these molecules physically bend their targets over a molecular knee to lower energy barriers, speeding up reactions to 40 million times a second without burning us alive.
Which is still hard to believe, honestly.
And we ended up seeing how manipulating these machines governs everything from newborn screening tests and antifreeze antidotes to laundry detergent and COVID-19 treatments. So what does this all mean?
Well, if we connect this to the bigger picture, when you understand enzymes, you understand the actual gears turning the universe of living things. And I want to leave you with a thought that brings us right back to the beginning, to the discovery of those RNA ribozymes.
Okay.
If RNA can act as both the genetic blueprint and the enzymatic machine, it raises an incredible question for the future of medicine. Could we eventually engineer synthetic RNA ribozymes and inject them into patients to cure missing enzyme diseases like Tay-Sachs, completely bypassing the need for complex proteins?
Wow.
The very molecules that might have kickstarted life billions of years ago in the RNA world could be the ultimate medical hack of our future.
The deepest history of life becoming the cutting edge of modern medicine. Thank you for joining us on this deep dive. Next time you digest a meal or wash a shirt, take a second to appreciate the trillions of invisible, hyperfast molecular machines contorting and snapping bonds to make it all happen. Keep exploring, and we'll catch you on the next one.